We have two cell models. This is one of them. I call this one the small cell. You can find it in your lab manual on page 10. This model shows some things better than the other big model, and then the big model shows some things better than the small model, which is why we often have two models or even more for a given structure. So the one thing that this model doesn't show very well is the plasma membrane. But if you look over here, you will see the number 18 is supposed to represent the plasma membrane because it's on the edge of the cell. But you will find when you watch the video on the big cell model that that plasma membrane is clearer. So I usually use that one to ask you about the plasma membrane if I'm going to ask you about that. This is the nucleus. And if you look inside, you can see what kind of looks like hair, ball of hair, right? And then there's a sticker, number 3 on it. That represents DNA in the form of chromatin. The white balls inside the nucleus are nucleoli, plural. Nucleolus is singular. The nuclear envelope is the membrane around the outside of the nucleus. The nuclear pores are these holes. Now, what I wanted to say here is when you look at number 1, and I know you're not yet accustomed to the numbers on these models. This model's from Germany, and a lot of our models are. So that's a number 1, even though it looks like a 7 for us, but that's a number 1. So on a test, if I ask you what number is on something, you need to draw it the way that it is on the model so that there's no confusion. If you draw a number 7, then it's wrong because the number 7 is on a different structure and it looks different. Actually, it's right there, the tip of my finger is number 7. But back to the nucleus. So if you look at number 1 and number 2, you cannot tell whether number 1 is supposed to represent the entire nucleus, just the nuclear envelope, or a nuclear pore, which is this hole. Same with number 2, actually. So on the test, I would ask you which which number is on the membrane. No, I wouldn't even do that. That wouldn't be clear enough. I'm always going to be as clear as possible. So I would say what number is on the nuclear envelope, for example, or what number is on the nucleus, or what number is on a nuclear pore, and I would take either number 1 or number 2, but it's still pretty unclear. So the best way that I could ask you this on a test is to say, what is number 1? And then in parentheses, I would say the little hole, or I would say the entire membrane, or the entire round structure. Okay, so that is number 1 and number 2, and then number 3 is DNA in the form of chromatin. So DNA or chromatin are both equally correct. I guess I could just go in order, and number 4 is weird looking. It's right there on a nucleolus. And then number 5 is, I don't know where number 5 is. So I'm going to go to number 6. Here is number 6 up here. This is smooth endoplasmic reticulum. This is rough endoplasmic reticulum. The difference between the two is clearly the little red dots. The red dots are ribosomes. Rough ER, rough endoplasmic reticulum, do not use abbreviations on the test. Do not say rough ER on the test. Rough endoplasmic reticulum has ribosomes on it. Smooth endoplasmic reticulum doesn't. These are free-floating ribosomes, so ribosomes can be found either on the rough ER or free-floating in the cytoplasm, in the case of number 7. Number 8, right above it, this is a mitochondrion. All these orange things are mitochondria. And number 9 down here is the Golgi apparatus, also known as Golgi complex. Those two are equally correct. And I just want to look real quick at the list. Oh, cytoplasm and cytosol. So number 17 down here is supposed to represent either cytosol or cytoplasm, which is the cytosol plus the organelles. Again, I would have to be really specific on the test question in order for you to know exactly what I'm asking. If I say what is number 17, you could say either either cytoplasm or cytosol. Okay, that's it.